Adina. So welcome everyone to CC Colloquium for February 24th, 2021. We're very pleased to have a speaker talking to us about the Earth and climate change for a bit of a pivot from some of the past colloquia this term. And I also wanted to make a special announcement about next week. So next week, we're going to be continuing in our series that we do every semester of graduate student presentations. So Ed Bowie, John Christoph, and Gregory Vance are going to be giving short presentations at colloquia next week. So please make plans to be there. Don't miss that. And with that, I think there's no announcements from CC leadership other than that we have our community conversation, our monthly community conversation tomorrow on Zoom at noon. So please mark that on your calendar if it isn't already there to hear the latest and greatest and ask questions. There'll also be a brief interview with Karen Anderson, who is an expert on vaccines here at ASU. So you can get some of your vaccine questions answered. And so with that, I'm happy to turn it over to Hillary to introduce our speaker.
Thanks, Ariel. So it is my great pleasure to introduce Adina Paytan, a sister oceanographer, who is visiting us today from UC Santa Cruz. The usual sorts of introductory information: she has an undergraduate degree in geology, master's degrees in science education and earth sciences, and a PhD in oceanography. And her research group is interested in looking at a wide range of diverse systems using chemical and isotopic tracers to understand biogeochemical processes in the past and in the present. And in particular, they're interested in understanding kind of the human impacts on Earth systems. That's all stuff you could read from her vita. So I always ask our speakers a couple of questions that you guys won't know the answers to. And so in my usual way, my first question was tea versus coffee, and Adina punted. She said coffee in the morning and tea at night. Cats versus dogs, and she likes all animals but has cats as pets. And I asked whether she had any new hobbies as a result of the global pandemic. And she says that she now takes a break for walking and does 7-minute stretches, and that she can multitask a proposal and a Zoom meeting. So with that, it is my great pleasure to ask Adina to share her slides and tell us about corals on acid.
There we go. Can everybody see my slides?
Looks good.
Perfect. So thank you everyone for inviting me. I'm really excited to share my research with you all. And hopefully in the next year or in the future, I'll be able to come join you and visit in person. I'm going to talk about corals on acid or ocean acidification. And as you see in the title, I call it the other CO2 problem, and I'll explain why shortly. But before I go into explaining or going straight into my talk, I would like to not forget to acknowledge the people behind the work. And first of all, my 3 graduate students who did parts of this work as their PhD thesis, Anna, Elizabeth, and Helen, and a lot of collaborators, biologists, coral reef ecologists, mineralogists, geochemists, and the most important, my collaborators in Mexico who facilitated the ability to go do the field work over there, and a lot of funding agency each contributed a little bit of money to this effort. So what am I going to talk about today? First, I'll explain what is ocean acidification and how it works. I will spend a little bit of time explaining why we care about ocean acidification, where the research is today, and then dive into specifically my work from the field work in Mexico and the implication of this work to all of us. So just starting and going into, first of all, this slide that I hope, I'm actually sure you've all seen. This is, even though I'm talking about the ocean, I want to show you, first of all, a figure of atmospheric CO2 and how the record of atmospheric CO2 has changed over the past few decades. You can see an increase in atmospheric CO2. This is a record from Mauna Loa in Hawaii. And most of this increase in atmospheric CO2 is due to carbon or fossil fuel burning and since the Industrial Revolution. So I know you've all seen that before. It's called the Keeling curve. And we all also, as a community, understand the implications of increasing CO2 in the atmosphere. What we call climate change, which results in increasing temperature because CO2 is a greenhouse gas, melting of ice caps and therefore also sea level rise, changes in hurricane intensity, and so on. A lot of research focused specifically about climate change relevant to CO2, and that's something that as a scientific community we've been studying for quite some time. However, if we look at the record of CO2 emissions, which is the blue line here, calculated from fossil fuel burning, we know very well how many barrels of fossil fuel are consumed, and compare that to what is actually in the atmosphere, which is the dashed purple line, the record from Mauna Loa, you see that there is a difference in the amount released to the atmosphere and what is actually in the atmosphere. About 30% of the emitted CO2 to the atmosphere is missing. And that missing CO2 is absorbed primarily by the ocean. Some of it goes to an increase in forest and greenery, but a lot of it or most of it is dissolved in the ocean. And that's something that is very, very basic science, very basic chemistry, which is dissolution of gases that any inorganic chemistry class would have taught you. Any gas in the atmosphere will dissolve into the water it's interacting with in proportion to its concentration, and also depends on temperature and salinity and so on of the water. So this is not something that is very surprising, yet as a community, we haven't thought about the consequences of this CO2 that is now present in the ocean. And the reason for that is primarily because there's a lot of carbon in the ocean. And therefore, the amount that would be dissolved from the emissions or from the atmosphere was considered to be small and therefore potentially not relevant. However, one thing that as a community we neglected to think about is that the dissolution or the fraction of CO2 that dissolves into the ocean is not immediately spread and diluted throughout the ocean, but happens primarily in the surface ocean. And it may have implications to that, to the surface ocean. And here I want to show you what happens to this carbon dioxide. You can see the carbon dioxide in the atmosphere dissolving and becoming dissolved CO2 in seawater. It reacts with water to form carbonic acid, which then dissociates to bicarbonate, releasing protons, which then bind to carbonate ions in seawater. And the end result of that is that we have more carbonic acid, a little bit more bicarbonate, more protons, which is acidity, and less carbonate ions in seawater. And all of this together, because of this carbonate chemistry of seawater, may have implications to the environment or to living organisms in seawater. How do we know that that's happening? Actually, we do have direct evidence of this process because people have been monitoring not only the atmosphere, this purplish red curve here, which again shows atmospheric CO2, but also seawater. So people go regularly to various locations in the ocean and measure dissolved carbon dioxide in seawater as well as pH. And these results over here, you can see is from the Hawaii Ocean Time series. You can see that in parallel to the increase in atmospheric CO2, the partial pressure of CO2 in seawater also increases, and the pH of the ocean decreases as well. So this is from one side, but anywhere you go in the ocean, you will see similar trends and similar data with differences from site to site, as I said, because of differences in temperature and other processes, but the trend is global. And again, it's not very surprising. This is expected from gas solubility principles. Now, what can we do with this data? This is actual measurements, but if you look at the figure, you can see that we don't really have a lot of measurements. We've been monitoring the ocean maybe 20, 30 years and in very unique small number of sites. If we want to look at the impact of the whole ocean, we really have to use models or calculations to determine this distribution or the pH in the ocean or the difference in ocean pH over time. So this figure here, this is results of a calculation or a model that computes the change in sea surface pH between 1700 and 1990. Obviously, in 1700, we weren't able to go all over the ocean and measure directly the pH. So this is done using calculations based on records of temperature and records of how much CO2 is in the atmosphere. Simple, as I said, solubility, temperature dependent. You can calculate how much CO2 is being added to the ocean. And based on that, you can determine the pH of the ocean from the carbonate chemistry, and you can calculate the difference. So what's important here is that nowhere in this map is white. All of the data over here in the Caribbean is just because it's not enough data to calculate it, but everywhere in the ocean, you see that the numbers are negative, or the pH of the ocean is decreasing, and this is what we call ocean acidification. And it's decreasing by these numbers that look kind of small, 0.1 pH units. I just want to remind you that pH is measured on a logarithmic scale. So a 0.1 pH unit is actually a fairly large change in ocean acidification, or in the actual hydrogen ions or protons in the ocean. Now we can do the same exercise now. Instead of calculating what the pH in the ocean was in the past, like at 1875 or 1995, but now based on projections of how much CO2 will increase in the atmosphere in the future, we can also calculate the pH or expected pH. Business as usual, if we keep on the lifestyle we're living in now, for 2050 or 2100. And you can see here that the pH of the ocean over these 50 to 100 years have shifted, or we expect that it will shift from values around 8.2 or higher, definitely above 8, to values of 7.7 or 7.6. Now, again, those of you who remember in your basic chemistry, this is not acidic conditions. The ocean is not going to become acid. It's all still above pH 7, but the trend is towards acidification. So the pH of the ocean will be decreasing, as you can see here. And again, as I mentioned, this 0.3, 0.4 pH units is a very big difference in the ocean chemistry and ocean conditions. So with that in mind, the question is, what are the potential implications or impacts to marine organisms and ecosystems? And there's going to be a lot of things that pH is involved in, or carbonate ions are involved in, or the carbonate chemistry is involved in, that may be impacted. And I emphasize the word potential impacts because we don't know. This is an area of very active research. And in fact, if I'd shown you a plot of the number of papers per year on the topic of ocean acidification, you would see that this has been increasing exponentially as the community realized that this is going to be a problem and the funding agencies encourage such studies. So you can see that the impacts could be on reductions of calcification, changes in the lifespan or the reproductive potential of different organisms. It could be in the fitness and survival, food web structure, and even inorganic things like sound absorption that can have impacts on organisms like whales that communicate in the ocean, homing ability, and so on. So all of these are important impacts that are now being studied extensively. In this talk, I'm going to focus specifically on the impacts of ocean acidification on the process of calcification or forming calcium carbonate shells by organisms. And again, why is that important? If you see that figure that I've showed you before, when carbon dioxide is forming and all of these reactions, one of the consequences is that the protons react with carbonate ions, forming more bicarbonate, and therefore the carbonate ion concentration in seawater will decrease. And this carbonate ion is the major building block of organisms that form calcium carbonate shells, like you can see over here. Now, this figure over here shows you how carbonate ion concentrations have changed again from 1700 to 1990 based on the same set of calculations that uses atmospheric CO2 and sea surface temperature, assuming ocean calcium has not changed or alkalinity has not changed, and you can calculate. And again, you can see about 30% decrease in the content of carbonate ion concentration. It varies from place to place, but it's always a negative value, the delta or the change. So there's less carbonate ion concentrations. And that has, as I said, implications to organisms that need this carbonate ion to build their shells. And these organisms that are collectively called calcifiers, you can see come from various groups, very diverse groups, from coralline algae and foraminifera, corals that build the building blocks of coral reefs, crustaceans, mollusks, as well as, very important down here on the right side, tiny organisms like coccolithophores or pteropods that are part of the plankton and that are the base of the food chain. So if these organisms are not there, then the fish that depend on these organisms as their food source would decline, with implications to people, fisheries, and our livelihood as well. So there is a lot of implications for these effects on food webs and the organisms that depend on these carbonate ions for their survival. Again, I'm going to focus specifically on corals and coral reefs. And there are several reasons why my work focused on this specific ecosystem. First of all, it's an ecosystem with a large biodiversity. So it's really important in terms of the number of organisms that live in these coral reef environments. And this ecosystem also provides a lot of important ecosystem services to people. Anything from fisheries, nurseries for fisheries, protecting the coastal cities from big storm surges, and even a lot of medication is formed from natural products that are from reef organisms. In addition, most of the corals that form these reefs are made from a mineral, a calcium carbonate mineral that is called aragonite, that is more soluble than calcite. So we suspect it is going to be more susceptible to these low pH conditions or to acidification. And also the coral reefs, because they are abundant mostly close to shore, they're impacted by a lot of other stressors. Global stressors like global warming, which increases the likelihood for bleaching, as well as local stresses like nutrient and pollution and people like myself diving and disturbing this ecosystem. And finally, the coral itself is a relatively simple organism. It's basically a sac made of 3 layers of cells, and therefore it is expected that they'll be more transparent to the environment and particularly sensitive to external conditions. So there's lots of reasons why these ecosystems are important and potentially susceptible. And again, like the figures I've showed you before, you can calculate how much carbonate ion will change in seawater over time. Here I'm showing you not directly carbonate ion, but a function of carbonate ion that is called the aragonite saturation. It's basically the carbonate ion multiplied by the calcium ion activity in seawater, but because we assume calcium does not change much due to increased CO2, it's mostly reflects changes in carbonate ion availability, but it's translated to a variable that's directly important to the coral organism for calcification. If you look at the scale over here at the bottom, you can see that the red values of above 4 aragonite saturation are considered to be optimal for coral calcification. And in the 1700s, early 1800s, most of the area where coral reefs grow, these black dots that you can see here on this figure, the corals were bathed in water that had optimal conditions for their calcification. And with time, you can see here that the aragonite saturation decreases. In 2040, it's well expected to decrease in even more. 2100, it will move from levels of optimal through adequate to marginal, and then at 2100 to low. And on the aragonite saturation scale of 2.5 and below is considered very low or extremely low and very non-conducive for corals to calcify. And the reason we know what is optimal versus marginal is based on lab experiments where we see the conditions and the growth rates of corals under these various settings and conditions. So with that in mind, this caused a lot of concern in the media, anywhere from popular newspapers to magazines to scientific journals, even to, kids at school describing the coral reefs in 1950 and in 2020. Hopefully it's not really that dire. But obviously, because this is such an important ecosystem, people are concerned and worried that the ocean acidification will cause all the coral reefs to disappear, and there's a big coral crisis happening. As I said, most of this is based either on calculations of expected chemical change in the ocean and mostly lab experiment where corals are exposed to such conditions. So let me show you a few interesting results from these lab experiments or manipulation experiment. This figure is from Fine and Chernov, a paper published in 2007. And what they did is they took corals from the Red Sea and put them in like a flow-through system where they circulated water with lower pH. They pumped CO2 into the water to reduce the pH, and they watched what happens to the corals over time. As you can see here, that's how the corals started. And after a few weeks of circulating this low pH, again, when I say low pH, I don't mean really acidic, just lower than their natural environment, as expected by 2100. And you can see that the corals, the actual polyps of the corals did not die. They're there, but they became more like sea anemones. They stopped calcifying. So I actually was one of the reviewers for this paper, and I was asked to write a little commentary about the work. And what I said is that that's actually good news for corals, because the corals themselves are not disappearing, but it's bad news for coral reefs. And remember, the reef structure is actually what provides most of these ecosystem services and biodiversity, with all the niches and the sub-environments within that system. That also pointed me towards looking at the geological record, which I mentioned in that news and views. And if you can see here, this is the Scarlatinian coral or calcifying coral evolution, and they started existing in Earth and since then persisted for 450 million years. And over this time interval, there were quite a lot of episodes of extinction and low pH and so on. And we see that they kind of, there's contractions, they disappear, and then they grow up again. And it's possible that their ability to be naked, like you can see here, but not get extinct, is what caused them to disappear from the geological fossil record, but not completely from the environment, and then reappear again, because it's unlikely that that's just evolution again of the same species. Like you can see here, it was abundant, hardly found, and then abundant again. So that kind of tells us that maybe these corals can survive either as naked coral sea anemones, or that in some locations, a small number of these corals managed to survive and then they proliferated again. So that gives us a little bit of hope that maybe they won't all go extinct even in the future, and that they're actually fairly robust organisms. But keep in mind that was before people were around, and we're stressing this ecosystem a lot more. So some other data, this is nice work by Ann Cohen and her group, and you can see here again, this is a coral exposed to present day conditions, and then increasing the acidification or decreasing the pH to 2050, 2150, and so on. And you can see that the white parts here are the coral skeletons, and they're just dissolving away, similar to what we've seen with the naked corals from the previous study. But interestingly, if you look at the right figure, you can see that you can reduce the aragonite saturation from the optimal 4.5 conditions all the way to 2.5, and they're still calcifying happily. And then only past that point does the calcification decrease. And again, this is exactly why we consider this 2.5 as suboptimal for corals calcification in nature. But if you keep on looking at results from the literature, you can see that it's a lot more complicated. So here you can see again, normal pH or PCO2, high PCO2, low pH. And if you just see the results at 25 degrees, you don't see really any impact of these two conditions. But with higher temperature, you do see a significant decrease in calcification. So in nature, where you have lots of things changing together, the lab experiment may not necessarily reflect the complexity of the environment. And particularly, the big question is, if, assuming we maintain corals healthy, we don't stress them and give them sufficient time, will they be able to acclimate or even adapt to this ocean acidification and maintain their high calcification rates and sustain the reef infrastructure and growth? To answer this question, you cannot take corals into the lab because they reproduce very slowly, so you can't look at their evolution over time. And also you're taking them out of their environment so they don't have their diseases and predators and symbiosis and all of the other ecosystem interactions. So in order to answer this question, you really have to go and make observations in the field. And the way to do that is to look at ecosystems or places where corals have been exposed to low pH conditions over long time intervals. And indeed, in 2011, a nice study by Katrina Fabricius and her team found or compared settings that in the Papua New Guinea reef area where there are these submarine vents or volcanic vents that spewed CO2. So these bubbles that you see here in the figure, these are CO2 vents in this volcanic region. The CO2 then dissolves in the water, causing the pH to be about 7.7 or less. And not far from these vents, there's areas where you have pHs of 8.1, and then you can compare the conditions here and here and try to see what are the differences. And this figure on the left shows you these differences. So when the ratio is 1, that means there's no difference between this condition and this condition. And these are the various properties they monitored. And you can see if you look at red calcareous algae, for example, or sponge cover, or structural corals, all of these things seem to be lower, so they're less abundant at the low pH. There are some things that are doing better at the low pH, and if you look here, these are the non-calcareous algae and seagrass cover. These don't calcify, they don't need the carbonate ions, they like CO2 because they photosynthesize, so they're doing better. But surprisingly, one other component that looks like it is doing even better is this massive porites. Massive porites are a coral, calcifying coral species. And if you look at over here, these mounds, white mounds that you see right there and here, these are calcifying corals and they're right near these vents and they seem to be growing fine. So that kind of gave people a pause to think about, well, how come these corals are surviving there? And one of the limitations of this study was that these vents are short-lived. Pressure builds up in these magma chambers, gas is released, you get this vent spewing CO2 for a few years, and then the pressure subsides, the CO2 vent disappear and they pop up elsewhere. So it's possible that these big coral heads were there from before that vent was there. And if the vent was here, they would not have settled. And maybe they're just there because, you know, they're barely sustaining and not growing. So this has some suggestions and some limitations, and that brings us to my study. My study looked at kind of a similar setting. However, instead of these volcanic vents, we were looking at places where the ocean pH and carbonate ion saturation is low because of discharge or flow of submarine groundwater. And that's in the Yucatan Peninsula. So if you look at this map here, Cancun is up here at north. This is about 20 minutes south of Cancun. And these squares show you locations of submarine springs. The Yucatan Peninsula is a karst system. That's a flat area where the rocks are made of calcium carbonate, and as rain falls on that region, instead of flowing to the ocean in rivers, it actually percolates into the ground and flows into the ocean in these surface caverns or cave systems. And then the water, this rainwater that has a lot of dissolved calcium carbonate and CO2 in it, discharges in these ojos. These springs are called ojos de agua. And you can see here why, because it looks like an eye on a column day. This in the back is like the hotels along the coast of the Yucatan Peninsula, and these submarines, ojos, discharge continuously because it's a groundwater system that has been there at least for 15,000 years at the minimum, because that was when sea level was lower, maybe during the last glacial period. But these caverns are there and have been there for a long time. And you can see this water discharging all the time at these vents. And this water brings to the coastal ocean, right at the reef, like you can see up here, low pH, low saturation waters that is seeping out there. Importantly, this water, before it discharges in the subsurface, in the groundwater, it mixes with seawater. So it's a mixture of seawater and that groundwater. And that's important because corals will not grow in fresh water. But the salinity of this water is very similar. It's just a little bit lower than their natural environment, and therefore they can still grow. So the idea here is, okay, let's go to these ojos and see what grows there and compare locations where you have these springs to nearby locations in the same area, just a few meters away, where you don't have these springs. First thing we did is we deployed sensors to make sure that the pH is always low and the discharge is continuous. And here at the bottom, you can see these are just days in a year. We had these instruments there for several years, collecting pH, temperature, and pressure and oxygen content of the water continuously every half an hour. The blurry part of this picture that you can see over here is not me shaking and not being able to focus, but actually shows you this different density water that is seeping in that area. So we put the sensors right at the seepage, and you can see here that the pH is quite low continuously, despite the large variability. The second, we also took other chemistry samples, alkalinity, dissolved in organic carbon, calcium, and so on, as well as salinity, as you can see over here. And again, you can see the discharge of the water. Using all of this chemistry data, we could calculate the saturation level for aragonite at this location. Second thing we did after we verified that the springs discharge undersaturated water continuously is we wanted to look at the biology and what lives there. For that, we deployed quadrats and did transects. So you can see here along the distance, every 25 centimeters, we put this square and we looked at everything that grew in these areas and did some kind of survey, along with, again, the chemistry of the water along these transects from the center of the hoho or the spring and away from there. And you can see here a summary of these results. This is a little bit of a complicated figure because I tried to put all of this data together. So at the top, you can see the numbers. This is just meters along the transect. So this transect up here was 12 meters long. This one was 3 meters long. And then the colors along these bands are the calculated aragonite saturation every 25 centimeters from discrete water samples we were taking. So you can see the saturation varies from 4.2 to less than 1 undersaturated water. And the distance between these areas that are undersaturated and the completely high saturation is just a few meters. So right here at 0.4, 4 meters away from this location, you can see water that has a saturation of 4.2 and water that is below 1. And this these areas, because the density of the water is lower than the surrounding, it doesn't spread laterally, but actually goes up as a plume to the surface. And that's why you see these fingerprints or ojos. Now, as you can see that, you can also see here this hole where the water is discharging from the low pH. And right here at these locations, you can see corals. So you can see corals growing right within this low pH plumes everywhere that you can, we looked. So this is kind of actually was very, very surprising, but also encouraging. So some corals at least can grow even in undersaturated conditions. And these corals were there since they settled as little larvae, and they were there and exposed to these conditions for their entire lifespan. So that gives us a lot of opportunity to look at these specific individual colonies and what's unique about them. So when you look at this data, it looks very promising, but it's not all that great, because if we compared the areas where the aragonite saturation was larger than 2.5 to those where it was less than 2.5, you can see that the number of species decreased significantly. There's about 10 different species outside that area in 2 meters away in the back reef lagoon, while right at the low pH, only 3 calcifying coral species were able to grow. So the diversity of the corals was much lower. The coral area covered was lower, and the number of individual colonies, at least for some of the species, decreased close to these vents. Moreover, the specific 3 species that grew there seemed to be very robust species, but not major contributors to the reef framework. So they grow, but they don't build reefs, at least in that location in the Caribbean. And so the framework of the reef may not be as robust as we think, and definitely the biodiversity of this, if this becomes the future, the reefs of the future will be lower. The other thing we did is we took coral cores and wanted to look at the impact of this acidification on the coral physiology, and specifically coral calcification. So we looked at calcification rate as well as other parameters that were suggested to impact calcification. After we took a core, we made sure to close it with cement to ensure that the corals grow over, and we did check and we didn't kill any corals there, so we're happy that we did not harm the system. So here you can see some of the results of these corals. You can see up here a coral collected from the low pH area, and down here from the higher pH area. And again, we grouped that we had quite a few corals, and we grouped them based on the saturation level. And you can see the extension, which is the centimeters of growth that a coral does per year based on these growth bands that you can see here. And you can see that above 2.5 and also between 1.5 and 2.5 saturation, the extension is maintained. But as you go much lower, they don't even grow much in extension. The density, or how much calcium carbonate per unit area is being deposited, you can see decreases quite rapidly once you go below this 2.5. Then if you multiply extension by density, you get the calcification rate, and you can see this gradual decrease in calcification. So even though these 3 coral species actually can grow and survive and have a net growth over time in the low pH setting, the amount of calcium carbonate they put into their skeleton per given time is lower. So their density is lower. What are the implications for that? As you can see here, the implications are that the corals or the reef is less robust. It's more susceptible to bioerosion and borrowing, and the volume is here. So if you have a big storm coming A coral like this that has all these holes and fractures will not survive as well as the coral on the right that is away from these low pH settings. So the robustness or the structure of the reef will decline, as you can see here. The other things we looked at is the tissue thickness. There was some suggestions that in order to compensate for low pH, the tissue area or the area where the tissue is present increases. We didn't see that. The tissue area, this one is the low acidification, this one is the control. The tissue thickness was the same. The clade of symbionts, so different symbionts that live with the corals can have different impacts, and it has been shown that certain clades of symbionts protect corals from changing in temperature and their sensitivity to temperature increases. We didn't see different clades, so the clades are the same at both locations. Nutrient levels, it was high, difficult to assess because they varied a lot, and it wasn't clear that we saw any statistically robust impact. The one thing that we did see a difference is the number of symbionts. So the low pH corals had more symbionts associated with them, potentially providing some more resources, energy to compensate for the need to put more energy into calcification. We also looked at recruitment. That is, what if we put tiles of these carbonate rocks and let them stay there for a year, would we see baby corals settling and growing? And you can see here, this is a tile that was deployed at the control site. This one is at low pH, and just looking at them, you can see that there is a big difference. But the major difference was not in the coral settlement. We didn't have many corals settle in either place. There were about a couple, 2 or 3 in each of these locations. So nothing that was statistically valid to report. But one thing that was very clear is that these coralline algae, all these pink colors that you see here, these are coralline algae. They were very abundant at the control, while in the low pH, they were not there at all. And all that you would see here is filamentous algae or green algae. And that was the largest effect. So both the diversity and the number of genera at the low pH declined with aragonite saturation, and also the total percent coverage of these algae declined significantly. So that is really important because research showed that these coralline algae provide a kind of environmental clues for settlement of coral larvae as well. We also looked at benthic foraminifera. These are also calcifying organisms. And if you look at the left side, you can see the absolute abundance or the number of living forams per centimeter square of sediment. The dark color is the low pH. You can see they're all, and the names are just different spring names. Regardless of the spring, there was always less abundance of these foraminifera in total. But if you look at the relative abundance of these symbiont-bearing foram was higher than the non-symbiont-bearing. Again, hinting towards this interaction with the symbionts, which are photosynthesizing, giving some advantage to the organism, possibly as a source of energy for calcification. The last thing we did is a transplantation experiment. And here we wanted to look at the genetic response of these specific corals to the conditions and to be able to say, or what is their prospect for adaptation. Basically what we did, we took corals of 3 species, 2 that are common in these reefs, the Porites astroioides and the Siderastria. And this upper one, the Porites porites, was a species of corals that isn't naturally found at these ojos. They only are found at the forereef and not naturally at the springs. So we took these from the reef, that one, and this one as well, from a high pH and from the springs, and then we transplanted them into either low pH or high pH settings in their natural environment. What that means is we drilled cores, we cut them into smaller nibbings or pieces, we numbered each one of them, took photographs of each one of them, colored them in alizarin red to make sure we know their starting conditions, and then put them back into their environment. And that's how it looks here. You can see each one of these corals randomly selected, put on these mesh with their little number. This is like number 60, and it's a Poritis, and it came from the reef, and now it's deployed here or there. You can see the pink color from the alizarin coating, and then we monitor them initially daily, weekly, then monthly for 2 years. So after 2 years, we went back and looked at the survival rate, and as you can see here, there's the 3 species, and this is the percent survival. So we know how many, and we had hundreds of these nubbings, so it's statistically significant, not just 1 or 2 samples. You can see that regardless of the species, the survival rate, both in the control and at the ojo, is pretty high. So even this Poritis, which is a species that does not naturally grow at these springs, had a 70% survival rate in the springs. Generally, the control was slightly better, but they're all amazingly survived pretty nice, at least for 2 years. So again, this is kind of tells us that it's possible for these corals somehow to grow even at these undersaturated conditions, provided they're healthy and can do that. But, and you can see here, we had the sensors right next to them, and we measured all of the environmental conditions. So one thing that was very clear, once after 2 years we harvested these things and we try to look at their growth rate, is that regardless of the species, in all cases at low pH, which is these values here, the density of the carbonate decreased. So the corals grew, but put less carbonate per unit time. So they were less dense, which is consistent with what we've seen in the coral cores. The linear extension sometimes was faster, most times it was lower, and therefore the calcification rate varied from species to species. So there's a lot of variability, but what is clear and consistent is the low density, or the less carbonate being deposited. They also had more symbionts, more chlorophyll, and more protein in the low pH settings. Finally, what we did is we extracted the DNA and sent them out for metagenome analysis. And here I'm going to areas that are way outside my comfort zone and expertise. But what you're seeing here is a heat map, and each line here represents a gene, and the colors represent if it was over or under expressed. And you can see this is one species that came from a control site, and the same species that originated from a spring, put both of them transplanted into a control site, and you can see that they're different. Some of the genes are different between these, actually each one of these columns is a single nubbing. So these are replicates. You can see they're different, suggesting that their life history impacts their ability to acclimate, adapt, and change their genetics. And here it's the same thing. Each one of these is a different coral nubbing. And these are now, we're comparing the same species from the same origin, but now transplanted to either the ojo or the control. And you can see there's lots of genes that differ from each other. So what do we do with all of that? Obviously, there is no way we can look at each and every one of these and compare them. So with that in mind, one thing that my student started doing, we discussed, okay, what do we look for in these gene expression. And the decision was, why don't we look at genes that are involved in calcification? Alkaline ATPase, ion channel genes, and so on. So when doing that, surprisingly, we found that these genes were not those that differed most between the low pH and high pH, or the origins. These seem to be similar. So the next thing we did is, okay, let's look for those that are the most consistent and most different and see what they are. And maybe in retrospect, it was not surprising, but we did not expect that. It looked like the genes that were most different are those that are involved in metabolism, like fatty acid metabolism and things like that. Again, hinting towards the requirement for increased energy resources in order to adapt and calcify. And interestingly, all of these genes are present in all corals. So potentially they could use or amplify or utilize these genes to allocate more energy towards calcification. Obviously, if they have limited amount of energy, this will come on the expense of other potential metabolic needs of these corals. So the less we stress them, the more likely they have the potential to overcome these conditions. So with that in mind, that brings us to the fact that, you know, there's things we can do. Most importantly, we can protect the corals. We can lower our carbon footprint, we can decrease the change that they're experiencing and lower the other potential stresses they're exposed to. But there's also other technological processes that could be useful, like transplanting them from one location to another. If they're not doing well, they can be, like you can see here in the photo, you can actually move little pieces of coral and plant them and they grow like a little garden, or here. Or you can even go be more sophisticated and do what is called assisted evolution, or you're crossbreeding different corals in order to increase their capacity to adapt and acclimate. And then you can, you know, go all the way to looking at their microbiome or hybridization and so on. I'm not necessarily, that's way beyond my expertise, and I'm not necessarily promoting that, but there are labs that are focused on using this technology in order to help the corals survive, and potentially we will still have at least some corals, maybe not as high biodiversity, but they will ideally not all go extinct. So with that, I just wanted to thank you all, remind you to protect our oceans for ourselves and for the future. And this is to remind me to thank my lab. This is actually already 3 years ago in Halloween, we were Life Aquatic that year. And with that, thank you all, and I'll be happy to answer questions.
Thank you very much. That was very nice and clear. Thanks. That was I really enjoyed that. We have some folks in the audience who are actually trying to develop various kinds of learning experiences around ocean acidification for a NASA project. So I know they really appreciated you laying that all out so clearly. I think Hillary is going to moderate some questions, and even though we're running a little late, usually we can squeeze in a few beyond the half hour. So Hillary, if you want to do that.
Absolutely. So thanks, Adina. That was really cool. I was just teaching carbonate chemistry to my geochemistry class last week, so it was very timely. We have a few questions in the Q&A, and the first two are just sort of practical questions. First one is, how can there be pH measurements of the ocean from the year 1700? So where does that data come from?
That is a very good question, and I kind of tried to explain it, but I probably went way too fast. So what you do have for the 1700 is record of CO2 in the atmosphere from ice cores. And you have temperature records of the water. And based on the solubility, the concentration of CO2 in the atmosphere at that time, the temperature of the ocean, you can calculate the solubility of CO2 and from there calculate the pH. So these are not direct measurements. These are calculations based on models and assumptions.
Great, thank you. Another question from Jessica Swan is, could you speak to direct impacts of acidification on the US coasts?
Well, you know, Hawaii is part of the US, US Virgin Island, Puerto Rico. We do have Guam. All of these are territory, and Florida. Now, Florida, there's a lot of other impacts on the coral reefs over there. They're further away from shore, but they are still impacted. There's a lot of research ongoing on the corals in Florida. These naturally, there's less calcifying corals around the Florida coast. They're more fan corals, or, you know, they're octahedral corals that don't form big reefs, but there are some reefs there, and there's a very extensive research. In fact, one of the labs that I showed some of the research in my last figure here, I think some of these here are from Mott Marine Lab in Florida, where they do this research on coral hybridization. So this is a global thing. And the more close the reef is to the coast, the more impact it is. The effect in Hawaii is more complex or more impacted than in Florida, just because of the distance. Mm-hmm. The reef itself is way further offshore in Florida than it is in Hawaii. But this is a global phenomenon, and it's seen everywhere. To be honest, right now, the thing or the environmental impact we're more worried about for corals is the warming that causes bleaching than the acidification. Because that's been going on for a while, we know a lot more about it, and it looks like there's reoccurring bleaching effects that are caused by warming.
Yeah, the corals are getting it a hard job, a hard life in lots of directions. We have one question from Steve Ruff, and I'm going to let him talk.
Yeah, thanks. So I was curious about I think you pronounce it Porites coral, and you showed examples of healthy and degraded degraded versions, and there was a porosity to them that I wondered if that's, no, is the other image?
This one.
Yeah, that one. So are those holes forming in panel C, are those forming after the thing is grown or during the growth?
That's a good question. Most of this porosity happens after. The density of the carbonate that is formed is lower, and therefore it's easier for parrotfish and all sorts of snails and bugs to burrow into that. So it's more burrowing.
Yeah. So it's exogenous. It's other creatures coming after it.
Yes. When it's alive within and it's covered by the coral polyp tissue, it's not exposed that much to the environment, but the tissue is only on the upper part of the coral, and the rest of it is then more susceptible to this burrowing because it's less dense.
Okay. Yeah, that makes sense. Thanks.
Do we have any other questions for Adina? One more question. Jordan asks, could we provide the corals with more calcium to help overcome the lower carbonate?
Unfortunately not. There's plenty of calcium in the ocean. It's not the limiting part for their calcification. They're not nearly utilizing this calcium. So we can add calcium, we can increase the saturation, but they still need the carbonate in order to precipitate the calcium carbonate.
Yep.
So the ingredient that is in the least supply is the limiting factor.
So in order to be respectful of everybody's time, let's give Adina a round of virtual applause. And if you have more questions, particularly for your student in the class, you'll have an opportunity to ask Adina those questions later.
Absolutely. And thank you all again for inviting me. And Hillary, I'll be happy to send you the PowerPoint if that's useful for the students.
Great. Thank you so much.
Thank you. Thanks very much, Adina. Really appreciated it.
Thanks. Bye,
And thank you, Hillary, for
Bye,
hosting.
everybody.
Nice to see everybody.